So this is DNA replication. In our first round, someone told me it was very right that this DNA replication occurs in S phase of the interphase. And secondly, this is semi-conservative. You know, this word is very much important. Semi means half. Conserved means conserved, kept same. So basically, it is semi-conservative means it is when two DNA molecules are made from one. Where each new DNA molecule has one old and one new strand. So this is why we call it semi-conservative replication. And if you want to see this through diagrams, it should be like this. This is DNA. This is so stupid is my drawing. इतना सिंपल सभी आप देखें ड्राइंग नहीं बनती वैसे सही तरह. तो बच्चों ये जो है ना ये एक DNA बन गया and this is another DNA. Sorry, one strand. And the other strand is new. You can see. So each DNA has one old and one new. So this is what we call semi-conservative replication. Did you get this idea? But why we call it semi-conservative? Because both new DNA have half old and half new. Right? Any problem? Dear friends, any problem?
No, sir.
We are going next.
अच्छा सर, जब हम बोलते हैं कि अनकंडेंस होगा तो इसका मतलब यही है ना कि मतलब अगर हम पुरानी बात करें तो अनकंडेंस होने के बाद डीएनए के स्ट्रैंड वो होंगे ना.
I did not say uncondense, unwind. बहुत ज्यादा इजी है.
Alright.
Because uncondensing की डिटेल में थोड़ा और भी चीजें आ जाती हैं. Super coiling, इसका सारी. So better we should say uncoil. For example, देखो, this is DNA. It got uncondensed, uncoil, unwind. Unwind is a suitable word, वैसे. Because uncondensed is when you need to talk about stones and other things. So just think of the two strands of DNA. So dear students, this one is say 3' to 5', and the other 5' to 3'. I just keep speaking, and I actually don't know that whether everyone is understanding or not, because when I ask you question, no one responds. No one asking me question if something is tough. How would I know कि यार तुम्हें समझ आ रहा है नहीं आ रहा? But anyway, चलो. So DNA got separated. Hydrogen bond breakdown.
सर ये 3' और 5' का क्या मतलब है?
लो जी ज़ैनब बेटा, ये पहले मीटिंग में जो हमने पढ़ा था कि two strands go anti-parallel. Were you there?
Yes, sir.
So बच्चों, if I start briefing you 3' to 5', it will take me one full class, by the way. You know, shortly मैं आपको बता दूं कि two strands of DNA go anti-parallel because when base pairing occurs, their nitrogenous bases are not opposite to each other. So the other strand will have to invert. ज़ैनब समझ आ रही कुछ?
सर आपने वो रिकॉर्ड किया है, आप वर्क ग्रुप में भेज दीजिएगा.
हाँ.
जिसको नहीं समझ आए वो दोबारा देखें.
हाँ जैनब वो अभी वो सारा करेंगे ना, लीजिए पूरी सेशन खराब हो जाएगी। तो 3 prime to 5 prime discussion was this was the only topic mainly that we discussed anyway. 3 prime to 5 prime means one strand will have 3 carbon free of the first nucleotide and 5 carbon free of the last. And 5 to 3 means first nucleotide has 5 carbon free unattached and the last nucleotide has 3 carbon free. So they go separate, they are unwinding. And this place is called replication fork. This place is called replication fork. Consider this a zip, just like zip of a jacket. So the point where it is opening, it goes further to open. So replication fork. Now, new DNA chain will be made because look, if there is A, obviously T, G, A, C, whatever, and they got separated. So this is A, C, D, G, and so on. Now look, there are nucleotides present in the nucleus. So a nucleotide containing T came up. Imagine. Then A came up. Then C came up. Then G came up. Then T came up. Then G came up. And imagine these are the nucleotides. So will someone tell me that, look, nucleotides joined by hydrogen bonds, but their dash bond is not yet made. Anyone?
Phosphodiester.
Impressive. Hafsa mein bata rahi hai ki backbone is not completed. See, phosphodiester needs to be completed. So look, this is how it continues. So will someone tell me please, what is this? This new strand, should this be 3 prime to 5 prime or 5 prime to 3 prime? Hello?
5 prime to 3 prime.
5 to 3.
Yeah, this one is 5 prime to 3 prime. So this grows towards the replication fork. This strand is called leading strand. Leading strand. This is called continuous strand. Continuous strand. It grows towards replication fork. And in this chain, the enzyme that helps make phosphodiester bonds is DNA polymerase. DNA polymerase catalyzes. अच्छा Zainab, please आप मोबाइल यूज करना बंद कर दें, मुझे अभी ख्याल आ गया, because you use this in classroom as well, तो maybe ये मसला हो. DNA polymerase catalyzes making of. बच्चों, when I teach faster, you'll be thinking कि मैं जल्दी करा रहा हूँ. When I teach slow, you'll be thinking कि ये slow pace है. So please do stop me if something bothers you, so that I could explain you again. I'll be back in a minute. Sahi Mohammed Aki. I knew this. Anyway, so DNA polymerase catalyzes making of dash bond. Anyone? Oh my God.
Phosphodiester bonds.
Yes, and leading strand. Phosphodiester bond, COPOC. So this is all the package. Now the issue is new growth. But it's very important to remember. I just write a note here. New synthesis is always from 5 to 3. 5 prime to 3 prime. The new chain will be made like this. Now, can you please tell me that?
अगर ये जो लेफ्ट साइड पर है, अगर ये डाउनवर्ड्स से शुरू होके अपवर्ड्स बनता जा रहा है, तो दूसरा वाला अपवर्ड्स से शुरू होके डाउनवर्ड्स बनता जाएगा.
डाउनवर्ड बनेगा. Very good. Because नकी हमें बता रहे हैं कि it will be like this. See this one, 5 prime to 3 prime. Then when DNA will open a little more, then another fragment, 5 prime to 3 prime. Then another fragment, 5 prime to 3 prime. So I mean, then it opens and goes behind. So ये जो उल्टा बन रहा है ना, this strand is called what? Lagging strand. The one which is made away from replication 4. So this strand will ultimately be called as lagging. It is lagging strand. If I ask you what is lagging strand, so I'm asking the one which is made against dash. Anyone? Naki aapne nahi batana. Made against dash.
Replication 4.
No, I mean made against 5 prime to 3
A
prime,
leading
3 prime
strand.
to 5 prime.
5 prime to 3 prime. Or 3 to 5 अगर आप opposite जा रहे हैं.
Well done, बच्चों. ये made against 5 prime to 3 prime. Look, new census is 5 to 3 ना?
इसमें ना कि जो बन रहा है वो 5 से 3 की तरफ होगा. मतलब पुराने वाले के 5 से 3 की तरफ नहीं.
हाँ, मतलब बच्चों देखो बात ये कि this one is 3 to 5, तो इसके सामने बनेगा 5 to 3. It will be straight going up, but the next one on the right is 5 to 3, so you cannot have another 5 to 3 because two strands must be anti-parallel. So the one which is made against 5 prime to 3 prime is made in the brackets and the pieces. So lagging is the one which is made against 5 prime to 3 prime. And this is lagging strand. Lagging is the one which is made away from replication 4. And it is made in fragments called Okazaki fragments. Okazaki was the Japanese scientist. And but you know why fragments? Because DNA is not fully opened yet. And you know there is an enzyme which makes phosphodiester bonds between the fragments. And this enzyme is called DNA ligase. So DNA ligase, mobile shops. DNA ligase makes co-POC, या phosphodiester bond. या ये कैसी क्लास है, इसमें आपकी पार्टिसिपेशन? बच्चों समझ आ रही है सबको? प्लीज मुझे ये तो बता दो. Those who are not participating, do you understand what we are teaching you?
Yes, sir.
Well done. So DNA ligase makes phosphodiester bond between fragments. I mean Okazaki fragments. So the two enzymes are important: DNA ligase, DNA polymerase. And this is how two DNAs are made. And now I'll ask question, and I request Hafsa and Naki. ये नहीं बताएंगे और हमें बताएंगे ज़ैनब, नवाल और ईमान. ठीक है? And my question here is now appearing before you in purple color. And the question is, during DNA replication, dash double-stranded DNA molecules are made. Anyone? No, not Naki and Hafsa. Zainab, Iman, and Nawal.
Is it 2?
Two double-stranded DNA molecules are made. Okay, Zainab and Nawal. Hydrogen bonds are made dash and broken dash. Anyone from Nawal and Zainab? I mean, you have option once, twice. You use these words, once and twice. Yeah, twice and once. Anyone?
Made twice.
Twice.
They are made twice. और Zainab आप बताइए broken कितनी बार है?
Once.
Yes, because when DNA unwinds, hydrogen bonds break. So this two double-stranded DNA molecule. And now this is open to anyone. That replication is all along the length, yes, certain places. Hello? बच्चों, it is all along the
Certain place.
हाँ, वो ट्रांसक्रिप्शन होती है. रेप्लिकेशन में आपने 2 DNA बनाने होते हैं. तो this is all along the length. And at times कभी-कभी क्या होता है, dash are left unreplicated. ईमान बताएंगी, ends या middles?
The ends.
Ends. मुझे पता था आप बताएंगी और मुझे बाकी कोई बच्चे बताएंगे कि what are those ends called? Yes,
Telomeres.
wow,
Telomeres.
telomeres. Very good, telomeres. And those ends which are left unreplicated, they are replicated by what?
The enzyme telomerase.
Telomerase. Very good. So this goes all along the length. Basically, replication must go all along the length. And last question before we end today's session, and I'm looking for, no, I shouldn't say brilliant because everyone is brilliant. अच्छा, template पता है क्या होता है बच्चों? Template means guide. Template is what? Please, can you say with me? Guide. Template is the masterpiece. Template means the guide. Template means the code of instruction. So in DNA replication, do you think both strands act as template? Yeah, one strand acts as template.
One is enough क्योंकि
One
we
strand.
already know
Oh
कि
my
दूसरी
God,
साइड पर
no.
क्या होगा.
No, both strands act as template. Look, देखो इसके सामने C, A के सामने T आ गया. I mean, who decides that which base will come? T. A. T के सामने A आ गया. So I mean, new bases.
Sir, पर जब एक साइड पे A है तो don't we already know कि दूसरी साइड पर T ही होगा.
No, I mean, 2 new strands are made now. So against this 5' to 3' is also a new strand made. Against 3' to 5' is also a new strand made. So it means both strands act as template. Template is one against which a new strand is made. So बच्चों, new strand is made against one template या both one strand या both strands?
Both.
Both strands.
So both strands act as template. I repeat, DNA ligase makes phosphodiester bond between the Okazaki fragments, and DNA polymerase makes the phosphodiester between growing chain and the incoming nucleotide. Hydrogen bonds are made by themselves. DNA replication में क्या होता है कि you have 2 double-stranded DNA from 1 double-stranded DNA. So initially DNA unwinds, then hydrogen bonds break, then hydrogen bonds are made against both strands. The DNA polymerase helps make phosphodiester bond in the leading strand, and DNA ligase helps make phosphodiester bond in the lagging strand. Leading is also called continuous, and it grows towards replication 4, and it grows 5' to 3'. Lagging is discontinuous. It's away from replication 4, and obviously this is made against 5' to 3'. And this is end of the news. Bye-bye.